Hey everybody. So we are taking a short jump in the textbook this week. We are going to skip over chapter 12 and we're going to come back for it later. So we haven't rounded out outcome 1 yet, but we are getting into outcome 2 so that we can complete our second SAC next week. So looking at our content related to outcome 2, we are starting in chapter 13 this week. And we will look at a couple of select pieces of chapter 14 in the next couple of weeks as well. So outcome 2, having looked at evolution, and we now know a lot about DNA and how mutations can occur and lead to new alleles, we're interested now in outcome 2 in the technological applications of the things that we have learned in the biological world. So how DNA works, how enzymes work, and how mutations occur. What technology have we developed to harness those powers? So looking at 4 specific cases in chapter 13. So we are focusing on this set of dot points first, and we're looking at what we refer to as DNA manipulation. So how can we use our technology and our knowledge to manipulate DNA for our own means and ends? So we'll first look at a set of enzymes that we call endonucleases, and these are enzymes that are going to be very useful in both of the following technologies. And then as part of looking at these technologies, we'll look at other enzymes called ligases, and polymerases we're already familiar with. So take a pause for a second and see if you can remember the term polymerase and where have we heard it, what is it responsible for, what do those enzymes do? Okay, and then we're going to look at a technology called polymerase chain reaction, or PCR. Followed by looking at gel electrophoresis and the steps and applications involved in that, before finally looking at how we can transform bacterial cells with genes of our own choosing. And this is referred to as a method of gene cloning. All right, so looking at our chapter 13 PowerPoint, and it is those 4 distinct sort of areas that we're looking at here. Just looked at our key knowledge. So endonucleases first. Endonucleases are a set of enzymes that have been isolated in bacteria. So I think I have mentioned them before. I'm just going to put me somewhere down here. I think I've mentioned them before, but they go by 2 names. So we can call them endonucleases with that familiar "ase" suffix, creating that name of an enzyme, or we can call them restriction enzymes. So they go by both names. Okay, they are enzymes that pretty much what they do is they cut DNA. So they recognize a specific sequence and they will cut a section of DNA when they read that sequence along the DNA strand. Feature of them is that they are palindromic, which means that the recognition sequence that they are recognizing is palindromic. And if you look up here, you can see what I mean. So we've got the sequence GAATTC. That is the recognition sequence for our restriction enzyme EcoR1. And if we were to read that backwards on the opposite strand, it reads the same, GAATTC. So that's what it means by palindromic. So the sequence that the restriction enzyme will recognize is a palindrome. It reads the same in the opposite direction on the opposite strand. And so what the restriction enzyme does is, moving along the sequence, when it encounters this exact sequence of bases, it will cut the DNA. And I just said that we've isolated these enzymes in bacteria. So they are a naturally occurring enzyme, and the names that they are given are given because they're named after the kind of bacteria that they were isolated in. So, for example, Echo R1 was found in an E. coli strain, so it gets the name Echo for E. coli. And you see other names, Hay3, there's BAMH1. There's lots of these shortened, abbreviated names for these restriction enzymes, but all the names originate from the bacteria that they were found in. Why do bacteria have restriction enzymes? It's kind of like the bacteria's immune system. And I think this is where I might have mentioned them earlier in the course when we were looking at um, how bacterial cells can become infected, and you might remember that there's a specific kind of virus that can infect bacterial cells, and we know already that viruses will infect their host cell by injecting their DNA into that cell. So a bacteriophage, which is the virus that can infect bacteria, they work the same way. They will inject their viral DNA into the bacteria, and you can see how something like a restriction enzyme would be handy here. The restriction enzyme that the bacteria has, it has a sequence, it's programmed for a sequence that is found in the virus that would routinely infect it. So when the restriction enzyme recognizes that sequence in the viral DNA being injected into the cell, it gets cut up by the restriction enzyme. So that's their purpose in the natural world. What are we using them for? Endonucleases are a real tool for biologists. So we use it in cutting and pasting DNA, something called genetic fingerprinting, and identifying polymorphism. So I'll talk about those examples in a minute, but bearing in mind here that looking at our key knowledge and how it's changed for 2020, there is less of a focus on the actual applications of endonucleases, for example, in genetic fingerprinting. So we are more worried about the identifying what these enzymes are, how they work, and less about what they are actually used for. But we will still cover it just as a basic understanding. But hopefully we're doing the right thing there. The other thing that I want to point out about restriction enzymes is that there's two variations. There's lots and lots of different restriction enzymes, but they work in two different ways. There's two categories. They are either sticky ends or blunt end cuts. And I've got both of them in this diagram sitting here. So a sticky end is where in the recognition sequence, the cut that's made by the restriction enzyme is made in a staggered pattern. So you can see it's cutting between the G and the A, the first two letters of that recognition sequence, and on the other side in the same place. So it's a staggered cut, which means that when the DNA actually separates, you have this overhanging part here, which we refer to as a sticky end. This sequence though, so the recognition sequence for this restriction enzyme, GGCC, and on the other side, GGCC, its cut is being made straight through the middle of that recognition sequence. So that means when the DNA separates, there is no overhanging sticky end. The thing to realize about this is that it makes one or the other more useful when we're doing certain things with our DNA after we've cut it. Once we've cut a blunt end, there are ways that the backbone can be reformed. So it's not always true that you can't reattach this piece of DNA, but it's much harder and it's not ideal. And for the purpose of this course, regard this as being an end that you can't reattach. So if we've got a blunt end cut made, that DNA is not going to be cut and pasted into something else. Whereas if we've got a sticky end, if we then have another piece of DNA that has the same corresponding sequence, when they come in close proximity, those bonds can be reformed, and it will, or it can, what we call ligate back together. It can reattach. And that reattachment is done by another enzyme, that we looked at in the key knowledge. Its name is a ligase. So a ligase is an enzyme with the suffix ase, obviously, that can ligate or glue together, reattach these bases on the sticky end. When would you find two sequences that have corresponding or matching sticky ends? When they've been cut with the same restriction enzyme. So if we were to take two pieces of DNA, cut them with the same restriction enzyme, and they both have the recognition sequences, they would have matching sticky ends that can then be adhered together, ligated together. If it was cut with a different restriction enzyme with a different recognition sequence, you can imagine that those two overhanging pieces, they wouldn't match, and so you wouldn't be able to get them to pair up and ligate. Okay, so one of the examples that I had on the previous screen, was a good place, is identifying polymorphisms. And I've got the question mark here because I'm not entirely sure if we need to go into the details of applications of restriction enzymes and of gel electrophoresis. I'll just quickly show you why. So knowing the technique itself is still in the study design. So looking at enzymes and what they do is still there. However, when we look, and this comes up a bit more in chapter 14, techniques that apply DNA knowledge, specifically gene cloning, genetic screening, and DNA profiling has been eliminated. Genetic screening that does encompass identifying polymorphisms, and DNA profiling I'll come up to later as well. So I think it has been removed from the course, but I feel like it's one of those grey area ones where I want to give you just the base knowledge anyway, and not harp on too much about it, because I don't think it's going to be assessed this year. But I'm just pointing out why I'm saying that and why I'm a little bit uncertain. So a polymorphism that we can use a restriction enzyme to identify would be a case like this where there is a change to this base. So for our normal phenotype, we're looking at sickle cell anemia, which we're familiar with from looking at it in one of our previous practical activities. But it's due to this mutation from an A to a T in the DNA, and how that flows through to give us a changed amino acid at the end. And pop quiz, what kind of mutation would that be if we've got a changed amino acid? So it's a point mutation resulting in an alternate amino acid being added, and so that would be a missense mutation. Hopefully you got that. But the nice thing about this one is that the mutation is actually in a, I can't remember if it's the right way around, but I think it's in the middle of a restriction enzyme recognition sequence. And so if we mutate it, it's going to cut normally and not cut in the sickle cell version. When you read about this in the textbook, I might have that the other way around, but you can correct me if I am wrong there. But one or the other will cut with the restriction enzyme and the other won't. So you'll have a situation where if somebody has sickle cell anemia, they would have their DNA either cut or not, based on whether the restriction enzyme can recognize that sequence anymore. So a polymorphism just means the different alleles, a change to an allele, and we can use restriction enzymes there if we're lucky enough to have an enzyme, a restriction enzyme, an endonuclease, that can recognize that sequence. Okay, so another one of our big four, and the interactive lab that we're completing through the Melbourne University website is going to give you a trial run at looking at PCR and the steps involved in it. and looking at gel electrophoresis and bacterial transformations, all three of them in one experiment, to hopefully give you a bit more understanding of how these things work. But just to take you through the basics for PCR itself. So PCR is a technology that has been developed to amplify DNA. So it's used to create multiple copies of a DNA sequence. Why would we want to do that? Well, it's a form of gene cloning. So any gene that we want to clone and make lots of copies of, we can use PCR to do that. It may be a case where we've only got a small amount of DNA that's been collected and we need to amplify that to give us more to play with, to test. A great example of that would be for forensics. So at any crime scene, you may have only recovered a very small amount of the DNA that needs to be analysed, and to hopefully alleviate any pressure on the scientists that are doing that analysis and give them backup copies, because sometimes analysis doesn't go right. We would like to have more of that DNA to play with and work with. So PCR is basically a technique that's used to create lots and lots of copies of a DNA sequence. What you need to know is the parts that are involved in PCR, how it actually works, and the steps that it runs through to achieve this. So this is what we call a thermal cycler, and it's what PCR takes place in. So thermal cycler, because it's going to cycle through multiple temperatures that are required for each step of PCR to take place. Into this thermocycler, we will load our PCR tube. In the PCR tube, we have the following ingredients. A buffer. This is just a solution that everything else can be mixed in, and it has the right chemical components that will allow the reactions to take place. Within this solution, within this buffer, we obviously need to have our DNA sample. So this is our template DNA, we refer to it as. The template DNA will have already been sequenced, and things called primers will have been designed. And if I just refer to this side of the diagram, a primer is a short sequence of bases that is complementary to either side of our DNA, either end of the DNA that we want to replicate. I'll explain why that's important in a minute. Okay, so we've got our primers, that's our starting points. We need free nucleotides. If we're going to build new DNA, we need the building blocks of DNA. So these free nucleotides get loaded in. And then we get something called Taq DNA polymerase. So DNA polymerase is an enzyme that we've talked about before. And I said earlier in the recording, can you remember what a polymerase does? Polymerases, we've talked about two kinds, DNA polymerase and RNA polymerase. RNA polymerase, we've talked about when we're referring to transcription. So RNA polymerase is responsible for creating that RNA polymer, the RNA sequence from the DNA. Whereas DNA polymerase is responsible for replicating DNA, so creating a DNA copy of a DNA strand. So it's the enzyme that's responsible for facilitating the binding of our free nucleotides to our template DNA in order to form that new sequence. It's got a different name though, Taq DNA polymerase. So Taq is a shortened, and much like a restriction enzyme, it's a shortened abbreviation of the name of the bacteria that this enzyme was isolated from. I will talk more about why we need this specific enzyme in a minute. Okay, so we load all of these in. And then our thermocycler runs through 3 temperature settings that will allow PCR to take place. Okay, so number 1, the thermocycler goes straight up to its highest point, right up to 95 degrees. 95 degrees hopefully as a good biology student now you can appreciate that that is not ideal for our macromolecules. So enzymes will have unraveled and DNA will denature. That means that it will separate. So the bonds that are holding our bases together between the two strands, they break. And we end up with single-stranded DNA. So when you are asked to go through the steps of PCR, that is what you need to detail. That denaturation happens at 95 degrees, which results in single-stranded DNA forming from our double-stranded template. Okay, the thermocycler then comes down to 55 degrees. This temperature is a bit contentious, so be aware. 55 is a nice number to have in mind. Some texts will say 50 to 60, some texts will say 50 degrees. As long as you hit a number that is within the range of 50 to 60 degrees, VCAR is going to say that you're right. So 55 degrees is a perfectly fine number to have in mind and use. So the second temperature, 55 degrees, is for our second step, which is called annealing. This is where our primers that I talked about earlier bind to or join with their complementary sequences on either side of the DNA. Primers are necessary because free nucleotides and the DNA polymerase can't start adding bases where there's nothing to join to. So we need a starting sequence. We need a free end of the new strand of DNA for the nucleotides to attach to. So primers are necessary. So our primers attach at 55 degrees. Then we hit a very specific temperature, 72 degrees Celsius. And this part is called extension. This is where our Taq DNA polymerase is going to kick into gear. 72 degrees is the optimal temperature for our Taq polymerase to function. So it will start facilitating that binding of the new nucleotides to either side of the template. And by the end of this stage, we'll have two fully formed copies of our original template DNA. Now, just while we're on 72 degrees, we'll come back to the specifics of Taq polymerase. Why do you think we need this specialized enzyme? Remember, it's gone in at the start and we've cycled through all of these temperatures and we've landed on 72, which is the optimal temperature for Taq polymerase to function. All right, it is a heat-resistant enzyme. It has a very high heat tolerance, which we need because if we're going to load it in at the start and crank it up to 95 degrees, our enzyme needs to survive that. And we know that a human DNA polymerase, for example, its likely optimal temperature is going to be somewhere in the realm of our body heat at 37 degrees. So it's not going to be much good at 95 degrees. So we have this special one, which I did say has been isolated from a bacteria, and that bacteria is found in underwater vents, which are kind of the really hot environments, really extreme heat environments. So that bacteria has evolved so that its enzymes can tolerate that high heat environment, which also explains why its optimal temperature is so high at 72 degrees. All right, so we've gotten to the end of our PCR cycle, but it's not enough. So at the end of one cycle, we've got 2 copies of our DNA. We need far many more copies of our DNA. So the thermocycler will repeat that set of temperatures over and over and over again until we have a usable amount of DNA. So you could always add on that 4th step there. And if you were in exam and you had a 4-mark question asking for the steps of PCR, the 4th mark is for saying repeat. That cycle, that set of temperatures will repeat multiple times. Okay, so that is how we're amplifying DNA. What we want to do with that amplified DNA then is something like gel electrophoresis. So this is where, if we've got enough DNA now, we can visualize it. You can't see DNA. We can sequence DNA, but we can't see it until we use something like gel electrophoresis. This might be a familiar process. It might be something that you have looked at in year 10 or in year 11, and if it's not, that's fine. I'll run through the steps now. So where PCR was a technique that was used to amplify DNA, gel electrophoresis is a technique that is used to separate DNA. So we have case a lot of the time where we would like to break DNA into parts and separate those parts and see how big they are. So basically, DNA is a slab of, gel electrophoresis. It is a slab of jelly. This is a jelly substance that is immersed in a water bath in a container, and an electric current is run through it. So the setup looks like this. This is literally just jelly, and it's porous, so it has tiny holes through the jelly that would allow the DNA to migrate through. And the bath is filled with a buffer solution that has it has salts and things that enable it to carry an electric current. And then the electrodes are connected at either end of the bath in this very specific pattern. So the positive end has to be down the far end from where we're going to put the DNA, and the negative end attached to the end with our DNA in. In our jelly, when the jelly's set, they put what's called a comb into it that allows, when it's set, that the comb gets removed, and then we have these indentations at one end of the gel. So this would be, well, it's what we call a well, but it's an actual hole in the jelly. And this is where we're going to place our DNA sample. Okay, so DNA will get loaded into this end of the gel. When it gets switched on, the current starts flowing through the buffer. The DNA gets attracted to the positive end. So there's two properties of DNA that allow gel electrophoresis to work. Number one, DNA is negatively charged, which means that it is drawn to the positive end of the gel electrophoresis bath. So that's why it moves through. And two, our pieces of DNA in this mixture are all different sizes. The smaller ones, the smaller a piece of DNA is, the more easily it can migrate through the gel. So we'll see our smaller pieces of DNA run further through the gel than our larger pieces which get left behind. So the way we can interpret a gel after we've run the electrophoresis is that smaller bands will be visualized further down the gel, and larger bands will remain closer to the wells. But we can't see the DNA yet, so as this is running, you won't be able to see the DNA very clearly. So a second step that is involved is actually visualizing the gel, and you will see this in the university lab. Mixed into this, this solution is something called ethidium bromide, or they refer to it in the online lab as something called red safe. They're actually two different chemicals, but they do the same job. So they're both chemicals that will bind to DNA, and when you shine a UV light over it, it fluoresces. So wherever we have DNA, that chemical, the ethidium bromide or the red safe, will have attached to the DNA band, and you'll be able to see it fluoresce under UV light. It's really impressive to see in person. And just looking at relatively where our smaller and larger bands are, that's not quite enough. So another step that we throw in with gel electrophoresis is we run something called a standard. So this, lane A, for example, might be our standard of DNA. A standard is a mixture of pieces of DNA that we know how big they are. So I, for example, know that this piece here is 100 bases long. It's just a number that I'm pulling off the top of my head. But this piece here is 100 bases long. So I know that the smallest band that runs in my standard is 100 bases long. And it's run to this position here. So what does that tell me about the band that's here in lane C? It is smaller. So it's run further through the gel, which means that it is a smaller piece of DNA, smaller than 100 bases. So we use the standard to work out the sizes of our unknown bands, and you'll get a chance to do that in the online lab. Okay, and so then some of the ways that we can use gel electrophoresis would be something like this. So the example we looked at earlier, and I think I described it right the first time around. So this would be visualizing that polymorphism. The normal version of the gene has the restriction site intact. So when that's run with, or digested, you call it, when it's digested with the endonuclease, it will cut. And the sickle cell allele doesn't have the restriction recognition site intact, so it's not recognized by the endonuclease and it doesn't cut. And so the way that then visualizes on gel electrophoresis is with the individual that has the normal allele, we see two bands because the DNA has been cut, and so that would result in two pieces. And the person that has sickle cell anemia, theirs hasn't been cut, and so it's one larger piece. We can tell that this band is larger, not only because it is near the wells, but we can compare it to the standard. So we can see that the intact piece of DNA is at the same position as the standard piece that is 400 base pairs long. And so we can estimate its size to be 400 base pairs. Whereas if it is cut with the restriction enzyme, it results in two smaller pieces because they've moved further along the gel. One that is 300 base pairs long and one that is 100 base pairs long. And if we were to do the math there, that 300 plus the 100 piece, if they were ligated back together, they would give us the 400 base pair piece of DNA. So the math works. So I'm nearly 100% sure that you're not going to be asked to do something like this, but gel electrophoresis can also be used for establishing paternity. So if we have DNA compared between father, son, daughter, son, sorry, father, mother, daughter, son, and an unborn, oh, sorry, it's not, this one's not establishing paternity. This one is looking at another genetic trait. So this is sort of like our fly example where we just gave you the data. Someone that has cystic fibrosis, someone who has two alleles, so cystic fibrosis and the normal version of the allele, they end up with the pattern where we can see both alleles showing on the gel electrophoresis. So one is larger than the other, and the mother as well there is also heterozygous, which means that when they have children, I can see that the pedigree here is showing me that the two parents have a son who has cystic fibrosis. So that tells me that the son here has two copies of the cystic fibrosis gene. And is the cystic fibrosis allele bigger or smaller than the normal allele, just based on what we know about gel electrophoresis? It must be smaller because it's run further down the gel. So he's got two copies of the cystic fibrosis allele, whereas daughter has inherited both normal copies, and we can then establish the unborn child. But this is an example of genetic screening, so I'm pretty sure we're in the clear here. This is the paternity example, sorry. So when we're matching up potential fathers to a child, you're looking for the missing sequences between So the mother, this band is accounted for in the mother. These purple bands are all accounted for in the mother. And we're looking for the sequences that don't match up with the mother to be present in the father. So that would make male 1 here, the match. And the example that I'm covering there is a forensic example. So DNA recovered at a crime scene gives us a genetic fingerprint when it's run on gel electrophoresis and would match with one of our suspects. I'm just skipping over this very quickly, just giving us a basic idea. Okay, and then the last step that you're going to experience in the online lab is looking at a bacterial transformation. So this is where we're using something called a plasmid to deliver a gene that we're interested in into bacteria that's going to do the cloning process for us. So both PCR and bacterial transformations are forms of gene cloning. So PCR is a clinical form, not using a real life situation, whereas bacterial transformations is the gene being cloned and produced in situ, in real life. So the way it works is we take the gene that we are interested in, so that's represented by this purple sequence here, and we cut open what's called a plasmid. So a plasmid, I wish I had a diagram handy to talk to. A plasmid is a piece of circular DNA that is found in bacteria. So bacteria have not only their large piece of circular DNA, but they also have these smaller rings of DNA called plasmids. And they're very useful to biologists for this exact reason. We can use them to put genes in and take genes out and deliver our genes into the bacteria. So this is a bacterial plasmid. We've got our isolated gene that we're interested in. We need to first create a linear plasmid by cutting the plasmid open at the recognition site. And you can see here the sticky ends match, which means that both our gene of interest and our plasmid have been cut with the same restriction enzyme. That allows then that plasmid to be inserted into, our gene to be inserted into our plasmid. And a ligase can be used to join those two fragments together. And so this results in what we call a recombinant plasmid. So that's our first step. We've created a recombinant plasmid by putting our gene into a bacterial plasmid. What follows then is trying to get our gene, our plasmid, our new recombinant plasmid into a living bacteria so that the cloning process can take place naturally. This summarizes the steps that we've undergone and I just described on the previous slide. And we're now to the point where we want to get this plasmid into our bacteria. So this happens through a shock treatment. And there's two kinds of shock treatment that are usually talked about, either a heat shock where the bacteria is mixed with the plasmid, the recombinant plasmids that we've created, and a heat shock is used, so a high temperature is used, and that triggers the bacteria to take up the recombinant plasmid. Also, an electric shock can be harnessed, can be used to do the same thing. So it's a shock tactic, which usually triggers the plasmid to be taken up into the bacteria. And there's a couple of points of hit and miss here. So we want to know if we've been successful. So we take our bacteria that we think has been what we call transformed with our plasmid, and we incubate it. So it will be spread onto an agar plate, much like we did when we looked at our bacterial growth practical, and it will be incubated at an ideal temperature and allowed to grow. And hopefully we get some colonies form, which again, from first-hand experience, you know what that looks like. And hopefully they have our gene of interest present in them. So a couple of checks need to happen. So the example that we're being given here is for insulin. So our gene of interest is insulin. just going to skip into your style for this week so I can use it to talk to, because I've got 2 diagrams in there that are useful here. Okay, so looking at our diagrams here, these represent the plasmid that we're using. So before we insert our insulin gene, we've already got a very important gene here, and that is a selective gene. This one is called ANT-R, so it's for antibiotic resistance. So our plasmid that we're using is already resistant to antibiotics. And we've put our insulin gene in there. The question that we want to answer then is, have we been successful in getting our plasmid into our bacteria? If the plasmid has successfully been taken up by the bacteria, because of our selective gene that was present on the plasmid, that bacteria will now be able to grow on a plate that contains antibiotics. So it stands to reason that if we put antibiotics into our plate, any bacterial colonies that form here will have the ANT-R gene and therefore have our gene of interest, the insulin gene. Because, and just through the natural way that bacteria grow by making copies of themselves, a process called binary fission, each bacteria that results from that original bacteria will also be carrying the plasmid and therefore also be carrying the insulin gene. You can leave it as simple as the gene is harvested, it is purified, and it is ready for use. So that is our basic way of gene cloning. So in this set of slides, you have learned about 2 methods of gene cloning, PCR and bacterial transformation, and a technique, gel electrophoresis, that is used to do some checks. All right, so hopefully that gives you a good coverage of the basics involved in these processes. Good luck.